all the parts of our country. We have received something around 241 abstracts, and I think you will see something around 200 presentations and 100 posters in this particular conference. We had 21 proposals for pre-conference workshop proposals. We have already done 10, and we have representations 5 different countries. So ICON probably we thought about is for India, but there are a lot of neighboring countries which are also showing their interest in this particular conference. And we have a mixed list of plenary speakers, and these are the managers, the policymakers, and the academicians. So I think this is the only place where you see a policymaker and a manager interacting with the scientists, real academicians who are trying to solve the conservation problems. And the ICON is also about the spotlight talks. Like, we have also brought people who are just joining the conservation careers and who are very young from the different parts of our country to come and tell and share their experiences when they are starting the conservation work in our country. And we also believe that we live in a developed nation and our students are not paid so well, as you all know that. So we decided that there are a lot of people who came forward. So we have started from ICON 1. This is the only conference which offers the equipment grants to the students. It's not a big grant, but it's the beginning to help the people to do their research well. Last year, we have offered 100 bursaries to the students to come to the conference. And this year, we are also offering 75 bursaries to the students to come for this particular conference. And this year, we have also started something new, which we call as an ICON Tech Bridge. We all know that there's a need of a technology. But sometimes, probably we oversell technology to try to save our forests. The conservation is a different ballgame. It needs people on the field. So we should be aware of what's the right mix of technology which we need. And there's a requirement that there's a feedback from ourselves to the people developing technologies, to give them a feedback, what is the requirement of the people who are there in the conservation. So the idea of Tech Bridge is to bring the people from technology side to listen to us and think about our requirements. And it's also to think about the technology side people and to think about what is their role when we are thinking about conservation. And we all are here because we have a great sponsors. So it is not possible to bring all you together and to manage and arrange this. And these are all our sponsors and partners who have been responsible for us. And at the end, ICON 2027. So we'll be meeting again after 2 years. And we have not yet decided the place, but in the next 2 days, we all will talk to you people and interact with you people and to decide where ICON 2027 should go. And thank you so much, and wish you a very happy and a great stay in WI. And I wish that you people will interact with your fellow colleagues and with all other people present in this conference to make this a great learning for all of us. Thank you so much.
Good morning, I'm Adya, and I will be introducing our first plenary speaker. Our first plenary speaker is Dr. Hema Somanathan, and she will be talking about the sensory and cognitive ecology of bees. Dr. Somanathan is a professor at IISER Trivandrum. She is an evolutionary ecologist whose research spans behavioral ecology, neuroethology, and sensory biology. She is recognized for her work on the sensory ecology of pollinators, particularly bees and bats. She also integrates the field-based ecological studies with controlled behavioral experiments to gain mechanistic insights into pollinator behavior and plant-animal interactions. She is also interested in collective behaviors in honeybees and social spiders. Over to you, Dr. Somnath.
Pointer. Oh. Thank you. A very good morning to everybody, and thank you for this opportunity to be here. I visited the Wildlife Institute of India many decades ago when I was a new PhD student, and after that I'm coming back now. So it's more than 2 decades that I'm coming back to the campus. Thank you very much to the organizing committee for inviting me here to give this talk. I'm very happy and grateful for this opportunity. So without further delays, I would like to plunge into my talk. So I am from the Indian Institute of Science, Education and Research in Trivandrum, and my lab is interested in pollination ecology. Work basically a lot with bees of various kinds, honeybees, wild bees and so on, and we are also interested in the behavior of spiders. So that's a side project in the lab. So this is my group of masters as well as PhD students as well as postdocs, as you can see over here, and we work mainly in the landscapes in the southern Western Ghats, but also in the Eastern Ghats in the last couple of years. So basically what we are interested in is this is not working. Okay. Hello? Am I audible? Yeah. So basically what, all of us have a shared history with bees because we directly benefit from bees. So they offer direct services in the form of pollination services of wild plants as well as crop plants. And therefore they are also very important for biodiversity as well as food security. So for a long time, for centuries, for thousands of years in fact, people have utilized the services of bees. And so what you can see here is an old rock painting from Spain, which actually shows a person trying to harvest honey. And so this is evidence that honey has been utilized as a nutritious food by humans for thousands of years. Yet another painting called The Man of Bicop, where you can see people harvesting honey from caves. This is on caves, and these are honeybee agents. Now here you can see is that when people actually formally started looking at the interactions between flowers and pollinators was first through the work of Sprengel. He was a German biologist who actually did a lot of very intricate work on flowers, describing floral morphology, features and so on, and trying to relate them to what kinds of pollinators would be visiting these flowers. So his work is very well known. However, it stayed in the background for a very long time till Charles Darwin actually went and verified some of his writings, and then suddenly, thereafter, Springle became a very famous figure. And you can see, if you visit the botanical garden in Berlin, you will see that there is a plaque in his honor, which actually, gives him this rightful position as being the first person who actually started a formal study into pollination. Now, Darwin, amongst many other things, was also interested in orchids, simply because they had these fascinating shapes and fragrances and so on. And so Darwin had a lot to say about the interactions between, or the specialized kind of interactions between orchids and their pollinators, and he wrote a lot, very, very extensively about orchids. So, but if you come forward to the modern times, it is a work of this one person, Karl von Frisch, who worked a lot with honeybees. He was an Austrian biologist. He did these very clever experiments where he actually would train bees to come to a feeder, and then he would test them experimentally. So not only was he a very good naturalist, but he also introduced experimental approaches to studying bees, their behavior, and so on. And through these studies, he proposed he made two very distinct discoveries. One is the waggle dance of the honeybee, which essentially is a form of social communication, okay, where they use a symbolic language in order to be able to tell their nestmates the location of the food. So subsequent work on the waggle dances have shown that the waggle dance codes for two kinds of information. One is it codes for distance to the food, and the other is the direction in which food is present, always with the reference point being the hive, because the dances, you can see, are performed on the hive surface, and the reference point is the sun. So the position of, with respect to the position of the sun, where is the food present, is the information that's contained in the waggle dances. Now, people have used this in order to try and understand how far bees go in various kinds of landscapes. So what you can see in the graph here below is you have distance on the x-axis, and you have the duration of the waggle dance on the y-axis, where the duration actually tells you what is the distance that a particular bee has traveled. So this particular experimental paradigm, it's a very simple paradigm, but it's something that's extensively used even till date in order to try and understand the foraging ecology of bees. Now, I will come back to the waggle dance. I'll revisit it later in my talk. So what you can see over here is the second discovery made by Karl von Frisch, for which, for both of these things, he got the Nobel Prize in 1973. And here, for the first time, he showed that insects are capable of color vision. Okay? So till then, we thought that it's the human, sort of capability to be able to distinguish colors. But through his experimental work, it was established that bees can see colors. So it was the first insect that was demonstrated to have color vision. Now, what you can see here is again a very, very simple, elegant experimental design, where he trained the bees to come to sucrose solution, which was provided with a particular color. And once the bees were trained, they would recognize, or they would associate the sucrose solution with a color. When they did that, then he tested them. And in tests, what he did was he would provide the trained blue color with a number of other shades of gray, some of it which matched the blue in intensity, some of them were lighter or darker than the blue. And if the bee, the question is that if the bee has learned the color, then it should actually visit the blue and not any of the other colors. So if it was really able, if it had true color vision, then it should go to the blue color and not visit any of the other colors. So using this paradigm, he very elegantly showed that bees are capable of color vision. Now, later work showed us that the color vision in bees is somewhat different from what we know in vertebrates or humans, for example. So we are able to see the blue, green, and the red regions of the spectra, whereas bees can see the UV, and they lack a red receptor. Okay, so they can see UV patterns in flowers, and many flowers we know have UV patterns, basically because they are addressing pollinators such as bees. Now, what you can see here is human colors, okay, as it looks to the human camera eye. You can see that these are the shapes and colors of a certain set of flowers. For a bee, which has a compound eye, the resolution of the eye is not as good as the resolution of the human eye. Okay? So they have very poor resolution, And the colors that they see are quite different from how we perceive or we see flowers. They're able to see UV patterns, which makes the flowers look very different for bees as compared to how we see them. Those patterns are not visible to us. Now, as I said, they have poor resolution of vision. What this essentially means is that even at a distance of 10 centimeters, we see a flower very clearly, but it appears quite blurred to a bee, which means that in order to recognize a flower by its color, they have to be actually very close to the flower. Okay? So this has consequences for the way bees visit flowers, their foraging behavior, and so on. Now, with all of this work by Colborne Fish, helped by the Nobel Prize and the limelight that came with that, there were two model bees that gained a lot of importance. One is, both of them are European. One is the European bumblebee, and the other is the Western honeybee. Okay? And all of these, both these bees became very important models for apiculture, for crop pollination, for ecology behavior, for conservation, also for neurobiology and physiology, because they became very important models because there was this very elegant paradigm that was established, training bees, testing what they saw, what they didn't see, what they perceived, and so on. So therefore, they became very important models like the Drosophila, okay, in terms of neurobiology, physiology, and so on. However, honeybees are not just about those Western species. There are many other species of honeybees in Asia, because honeybees originated in Asia, and yet we know very little about the other species of honeybees. So if you see here, this is the Western honeybee, about which there is a lot that's known, and it's also very, very commonly used in apiculture in the West, in Europe, and in North America. But these are the other species that are found in India. For example, you have the Eastern honeybee, which is Serrana, and then you have the Himalayan honeybee, Laboriosa. You have Apis dorsata, which is called the giant honeybee, and you have Apis florea, which is called the dwarf honeybee, and so on. So all of these are bees that are found in India. And the difference would be that most of Indian landscapes are serviced by wild pollinators, wild bees, unlike what's happening in Europe or in North America, where there is a large industry where bees are kept in farms and they're transported right across Europe or the US in order to service plants whenever there is a cropping season. So so the point here is that we barely know or rather that we know very little about these bees. And let's see what is it, why should we be studying these bees? There are a number of reasons why these bees are important. For example, in the tropics, the flora is very diverse and different from what we know in the temperates. The bee communities in the tropics are very speciose. The distribution of resources, spatially as well as temporally in the tropics, is very, very different from what happens in the temperate regions. The abiotic conditions are very different, and the ecosystem services are mainly through wild bees and not through managed bees in much of Asia, including India. And also in the current context of climate and habitat modification, it becomes very relevant to study bees. Now, two things about what people have been interested in trying to understand bees is, one is, how do they actually locate flowers? And today, from the wealth of information, we know that they use multiple cues. A flower is it's like an advertising billboard where there are a number of features there. There could be color, there could be shape, size, contrast. Flowers have temperature, which can be attractive to pollinators. They have patterns, they have odors, they have textures. And they also recently, a couple of years back, it was discovered that bees will actually respond to the electric fields around flowers. So they can use all of these cues at different at different points during their flower search behavior in order to locate flowers. Now, on the other hand, there is also the question as to how do large colonies of social bees actually forage efficiently, because they're not just collecting resources for themselves, but they have to feed the entire colony, and they also have to store food in the colonies for the developing brood larvae, for the queen, and so on. And so therefore, they have to be quite efficient in the way they do it. And they have certain strategies which helps them improve their foraging efficiency or to forage efficiently. And some of those I will be discussing today. So one of it is called an innate template or an innate preference for certain kinds of floral traits. The other is that they have very, very robust and rapid learning and memory in order to remember the features of flowers. And of course, social information, whereby information is transmitted much more efficiently than each forager having to discover resources on her own. Yeah. So therefore, let me just now for the rest of the talk, I'll take you through all of these ways in which how we know that bees forage efficiently by, sort of using some examples from the work that my students do. The first example is about contextual learning, okay? And we show that bees can learn context very well, and they can modify their preferences or likes according to the context. Now, how we did this is that we looked at this particular tree species called Catunaregum spinosa. It's present in the Western Ghats. And the interesting thing about this plant is that, like some other plants, the flowers change color from white to yellow. Okay, so the young flowers are white and they're very rewarding. They have lots of nectar. The older flowers turn yellow. Okay, now the hypothesis that stands in the literature about this sort of floral color change, where most angiosperm flowers will actually drop off once it's completed reproduction or post-fertility. But there are some species like this one here, where the older flowers are retained and they change color. And the hypothesis is that the older flowers are retained because they provide a much larger display, which is attractive to pollinators, whereas the younger flowers are rewarding and fertile, and so therefore they help to draw pollinators to the plant together by providing a larger display. So we did these field studies where we looked at who are the pollinators, and we saw that it's overwhelmingly bees, but also butterflies and flies. And we looked at the pollinator visits to the white, the intermediate pale yellow stage, and the bright yellow stage flowers. And we see that all these flowers are visited, especially the early or the younger flowers, but the older flowers are also visited. So that made us look into whether the older flowers are rewarding. And yes, we found out that when we tagged flowers using usual methods, we found that at 0, 12, and 24 hours, when we quantified the nectar content, we found that the white flowers and the intermediate flowers, but especially the older flowers, still continue to produce nectar, which means that the older flowers are not only helping to signal and attract pollinators to the plant, but they are also providing rewards. Now, next what we did is that we measured using bee color vision models. I won't get into the details of that, but it helps us to try and understand what is a bee actually seeing. Okay? From some basic neurobiological principles, we can actually understand what a color appears like to a bee. So that is what is represented here in this Cartesian space. And this is a color loci of the white flowers, the intermediate flowers, and the yellow flowers. And what this essentially means is that these colors are actually, at the two ends, the white and the bright yellow, are actually distinct colors to the bee, meaning that the bee can tell these as two separate or two distinct colors. The ones in between over here, they are probably making errors, and they're probably visiting all of them. For example, they can't make out from the white from these pale yellow colors, or they can't make out these pale yellow colors from the bright yellow colors. But at the two ends of these color loci, they can definitely see them as two different colors. Now what we did then is that we trained the bees, just like the Carl von Frisch experiments, where we got them from their natural hives to fly into our experimental setup, which is on the rooftop. And we trained them to enter into this vimase apparatus. So the bee is here, it enters the vimase. This is called the decision chamber. And once the bee is in the decision chamber, it can see and make a choice whether to go to the right or the left. And at the end of these arms, we provided, during the training phase, an array of artificial flowers, which are either white or yellow, those two ends of the spectra, equal numbers of white and yellow flowers, and then also pseudo-randomly place them on the left or the right, so that the bees are not going just to the left or to the right if they have a side bias. And then once the bees were trained, which they get trained very quickly, so then what we do is that during the tests, we provide one of the arms with a yellow dominant display, which is like 3:1 yellow is to white artificial flowers, or a white dominant display, which is the reverse, where the white is 3 and for 3:1 ratio of white and yellow flowers. Then we looked at, once a bee enters the decision point, whether she prefers to go to the yellow dominant or to the white dominant display. So these are the results. You can see that from the decision chamber, most of the bees choose the yellow dominant display from a distance. Okay? And then once they have entered the yellow dominant display, So then they start visiting the white flowers. Why? Because they've learned that the white is more rewarding. We have, sort of these artificial flowers. We have provided them with the same kinds of resources or the same quantities of resources as the natural flowers. So they learn very quickly that white is more rewarding, and so therefore they prefer to visit white. Okay, so this is the, and then so this is the first visit of the bees. You can see that most of the bees that were trained, though they enter the yellow dominant arms from a distance, they switch their preference and then they start visiting the white flowers predominantly. As they make visits, multiple visits, you can see that they start incorporating yellow flowers into their visits. Basically, they start encountering yellow flowers, they find that they're rewarding, and so they can also switch their preferences, or they can incorporate more yellow flowers as the visit progresses. There are a few bees that entered the white dominant display, but those results are not really relevant for this talk, so I'm not getting into that. So what do we learn from this? We learned that they can learn 2 color cues simultaneously, and that yellow acts as a long distance innately preferred. We didn't do any training for them to prefer yellow. So yellow is somehow a very conspicuous, attractive signal from the long distance. And once they're within the tree or within our setup, then they can switch their preference to white and prefer to visit white at which are more rewarding. So now, if you consider that a bee is a very, very tiny animal with a tiny brain, if you want to think about the size of a brain of a bee, it's like you take a safety pin and put a point, and that is the size of the brain. It has a few thousands of neurons. But this is quite a commendable feat that they can remember multiple things consequently and make decisions according to the context. The next kind of behavior that is displayed in bees is something that is called floral constancy. Again, there has been a lot of literature talking about memory constraints in bees because of the tiny brain size. They can't remember too many things simultaneously, and so therefore maybe they should specialize on, or remember few things at a time. So what we did, I'll just explain to you what we did. So we had the natural hives again, and then we trained the bees. Let us say that in a natural situation, they are going into a meadow which has different kinds of flowers. You can see that there are at least 3 different species of flowers here. So bees show this behavior where if they encounter a diversity of flowers, during any foraging trip, they would just specialize on one of those flowers, ignoring the others. So the reason why they do this is thought to be that by specializing temporarily in a foraging trip on one kind of flower, they can actually become more efficient at handling it, because flowers have complex morphologies and so on. And so therefore, this overall contributes to their efficiency, and it also, by temporary specialization, they have to remember the features of fewer flowers, and so all of this helps them in order to be efficient. So therefore, constancy, if you have a scenario like this, where there are multiple flowers and a bee just focuses or visits only one kind of flower, we would say that that bee is very, very highly constant. Whereas another bee which visits all the flowers that are available would show very low or no constancy, and this is somewhere in between where there is intermediate level of constancy. So we looked at constancy again by training bees from the wild colonies to come into our experimental setup, which they do very willingly. And then we train, once they enter this experimental setup, then we provide them with a training color, which is a neutral color. So we don't want them to learn any color, so therefore it's a neutral gray color. And then once they are trained to come in and out of this setup, then we provide them with a test array, which contains blue and yellow artificial flowers. Okay? These are filled with sucrose. Sucrose is the reward by which we can get them to do all these experiments. Otherwise, they are not motivated to do anything. So therefore, we provide them with sucrose. And then we looked at the preference. Now, what we found is that this is, you can see the graph here, the proportion of bees that visit blue and yellow, you can see that there is a very strong preference for blue. Okay? Now, what we have done in this experiment is that we have sort of erased their memory for colors by providing them during training with a gray color. Okay? And then what we see is a spontaneous preference for blue. Yeah? So this is the constancy index, which essentially tells you how constant a bee is to a particular flower color in this case. Now what we did is that we then said, let us reverse their preference. Okay? So what we did now is that we trained the bees to a yellow color, which they associated with the sucrose, and then we provided them with blue and yellow colors in the tests. So the hypothesis here is that they've been trained to yellow, they know that yellow is rewarding, so therefore they should go to yellow. So what we found is exactly that. So they had we had managed to override their preference for blue by training them to yellow, and you can see that very rapidly they switched their preference over here. Yeah, and they show very high constancy to yellow, which essentially says that this is a very useful behavior for a bee because it's all the time encountering resources in the field, and those resources are not constant over time, which means that there are blue flowers that are attractive today, but there are some other flowers, pink flowers, that are attractive tomorrow. So though they should show constancy and specialize, they should also be willing to. So this is something where, once they encounter a rewarding resource, they're also willing to switch their preference, and this helps them to survive in the wild, collecting nectar from and resources from all kinds of flowers. So what have we learned over here is that they do show high constancy, which is mediated by their innate color templates or innate color preferences, and they can also change their preferences very rapidly and become more explorative. Now, in the third kind of paradigm, I will talk about how bees learn from each other. Now, when we talk about social communication in bees, it's a very fascinating behavior. One is, we know through the waggle dances, they are conveying, it's like a symbolic language, and they are conveying information about where a particular resource is present so that the nestmates can go and find it very rapidly without having to search for it independently on their own. The other kind of learning is learning by observation, and there has been a wealth of studies on primates. We've heard Meva give talks about learning in primates and so on, but in invertebrates it's not that well studied. Okay, so now what we show here is that they can learn from each other, and this again, you can think about it as a way in which they can improve the efficiency with which they can forage. So what did we do for that? Again, we had wild colonies. We trained them to come into our experimental setup. The bee has to fly in through this chamber and then make choices inside this experimental arena. What did we provide during our experiments? We trained hornets, which are the natural predators of bees. Okay, these are the Vespa tropica, the large hornets, to come through this setup. And we provided 2 petri dishes, which are provided with sucrose. Okay, there were 2 petri dishes with sucrose. We allowed the hornet to settle and feed from one. Hornets also, they're not just carnivores, they also feed on nectar, so therefore they like sucrose. So what you can see here is that we train these hornets to feed from a sucrose feeder, and then we allowed the bee to enter the setup. And then we looked at the choice of the bee to try and see whether they would settle on the feeder that had the hornets or the other feeder that just had the sucrose. In a second set of experiments, instead of the hornets, we allowed conspecific bees from the same hives and from neighboring hives to settle on the feeder, and then we looked at the choice of arriving bees on whether they would settle on the bees that already had conspecifics or the sucrose with the sucrose feeder alone. So this is what our results look like, and you can see that with the hornets, the arriving bees, when they saw a live hornet feeding on the from the petri dish, they avoided that feeder and they went to the blank feeder, which contains only the sucrose solution. Of course, they would avoid their predators. With one live bee feeding, okay, they saw a conspecific feeding. They chose sort of equally between the feeder with the conspecific and the feeder with just the sucrose solution. But when we increased the number of conspecifics to 3 to 6 bees, which we have clubbed together over here, you see that they begin to prefer to go to the feeder where the conspecifics are already feeding and not to the blank feeder. So what this tells us is that they are learning from each other, and they prefer to go to the feeder where there are conspecifics that are feeding, because it's sort of like a risk-averse strategy, or it's a risk-averse behavior, because if they see the conspecifics feeding, so that must be rewarding, or it must be safe, and so therefore they go there to feed instead of going to the blank feeder. So this is evidence that they are actually learning from each other through observation, and making decisions or modifying their choices very instantaneously in this case. Now, you can think about this as something like if you go to, there are 2 restaurants side by side. One is there are lots of cars parked in front of one, and the other is empty. You would choose to go to the one which has a lot of cars parked there, simply because perhaps that's a better restaurant which serves better food. So what we've shown here, therefore, is that there are social cues that they are responding to, and there are predatory cues that they learn to avoid or they innately avoid. We can't distinguish between those two, whether it's innate or whether it is a learned behavior. And there also appears to be a threshold magnitude, which means that when it's just one live conspecific, they may go to either feeder. They don't really go to the feeder which has the conspecific. But when it comes to a certain magnitude of 3 to 4 bees, then they start responding to the crowd response, and they do go to the feeder that has conspecifics. Now, here is a waggle
Honeybees
dance.
have some fascinating abilities, among them being able to communicate by performing a unique dance. It informs hive mates where a newly discovered food source is located. Every cycle of this waggle dance roughly describes the shape of the figure 8. Let's rewind and look in more detail. The bee only waggles on a part of its route, the straight run, indicated here by the waved line. The secret lies in the direction of the straight run, or to be more precise, in the angle between the straight run and the perpendicular, which in this case is 90 degrees to the left. This tells the other bees that food is available 90 degrees to the left of the sun. If the angle is 60 degrees to the right, they'll be flying 60 degrees to the right of the sun.
So that is essentially a video which tells us that they use the sun as a reference point in order to tell the nestmates through the waggle dances where the location of food is in terms of distance as well as direction. So people have tried to see whether the waggle dance codes other kinds of information, but that's not the case. So this is another video from, where we have recorded it from the lab. And you can see here that there is a bee with pollen on the legs. And this dancing bee is being followed by other bees, which would then get some information about the location of resources. And then they would fly off in that direction in order to be able to find food. So what we have done is we have used this dance because we can't follow bees, we can't radio track bees, we can't, we lose sight of bees very quickly once they've taken off, but yet we are interested in knowing how far they go and how do they forage in a given environment. So therefore we've used these dances as a tool to try and understand how far the bees go in different kinds of landscapes. So the landscapes that we worked in are plantations, agricultural fields, even urban areas, as well as in forests. And the tool that we used is that we trained the bees to a feeder which is placed at a given distance that we know of, 100 meters, 500 meters, a kilometer, and so on. And then we mark the bees that arrive at the feeder, so we know the identity of each bees. And then when they come back to the hive, we record their waggle dances, which then we can decode the information about where the bee has been using those waggle dances. And this is what it looks like. For example, we can see that in these 4 different kinds of landscapes, this is the center is the hive location, the blue dots are the nectar foragers, and the yellow dots are the pollen foragers. And you can see that they are roughly going a distance less than 400 meters in order to search for nectar as well as pollen in these different landscapes. If you look at the maximum distance that is signaled through dances, you can see that in the forest it's about 1.2 kilometers. In rubber plantations, it's about 2.2 kilometers. In agricultural areas, it's about 1 kilometer. And in urban areas, it's about 800 meters. Now, we have some ideas about why these may be so variable between landscapes. It really depends on the quality and the quantity of resources that are available in these different landscapes. But the important point here that I'd like to make is that if you look at the eastern or the Indian honeybee, the median distance is about 317 meters, and the maximum distance signaled is about 1.3 kilometers. Whereas if you look at the western honeybee, they travel much larger in order to find food. And this indicates that there are differences, there are basic underlying differences in the way resources are distributed between European habitats and tropical habitats, which actually has an influence with respect to how far a bee goes, how she forages, what kinds of resources she gathers at what distance, and so on. So what we know from this work is that they have much shorter foraging distances than compared to the western bees, and the habitat quality seems to influence the maximum foraging distances that are flown from the hive. I'm not sure how much time I have left. Do you know? Yeah. Okay. I think I have about 5 minutes. So the last part of my talk is I would like to give you a flavor about nocturnal bees, right? So all of us know that bees are diurnal, but there are also some bees that are nocturnal. And these are interesting because we barely know what nocturnal bees do in terms of the pollination services that they render. Now, this is, I mean, a lot of bee families have nocturnal species, and so therefore, it becomes an interesting topic of study. And we have been looking at various reasons for why bees may have turned nocturnal, because this is evolutionarily a much more recent behavior. And there are several hypotheses connected with resource availability or competition or reduced predation during the night and so on that might have led to this transition to a nocturnal lifestyle in bees. So I won't get into the visual ecology part, but what I would like to sort of demonstrate here is that we have shown that they actually have excellent vision at night by doing a set of experiments in the forest where this is the hive of a bee, which is in the dead logs of trees. And we train the bees to go in and out of their nest by placing a landmark over the nest. And then once the bees have been trained to accept the landmark over the nest, then when the bee leaves the nest one evening, so we change the position of the landmark. And the idea being that if the bee has learned the landmark, then she should directly go towards the landmark when she comes back from a foraging trip. And what you see here is the flight path of a bee during a normal exit. And this is the flight path of a bee after the landmark has been displaced. And you can see the very confused behavior of the bees here, which shows that they have learned to associate the landmark at even those very low light conditions. We've done these experiments at various moon phases, for example, half moon, full moon, and so on. And we see that they are capable of finding the nest under those low light conditions, quite correctly without making any errors. And we have also seen that they do seem to possess certain visual adaptations, which I don't have the time to get into, which allows their eyes to become much more sensitive under low light conditions. For example, these are the photon gathering apparatus in the eye. They are called the rhabdoms. And these are much wider in the nocturnal bees, which essentially means that they can capture much more light during the night, which are not seen in the diurnal species. So the same thing you can see here, that this is the facultatively nocturnal honeybee, and you can see that their rhabdoms are much wider, which allows them to capture many more photons of light than the diurnal species. We've also shown that even though they are active at such low light levels, it's not that they lose color vision at night. So they are capable of seeing colors at night quite efficiently, and we have done these experiments where we again train them using the similar paradigm to distinguish between various colors, and we see that they learn the training yellow or the training blue quite reliably, and they can visit this irrespective of whether it's a full moon or a half moon and so on, so which shows us that they learn the colors very reliably. We repeated these experiments even with the honeybee, which is facultatively nocturnal, and we find similarly that they can see colors at up to half moon life intensities, and so therefore they haven't sacrificed their color vision, which essentially means that they can detect flowers at night using color. Now we also looked at their pollination networks to try and see whether the nocturnal bees were using a very different resource from the diurnal bees, and what we can see here is that that's not the case, simply because you can see that this is the nocturnal bee, and each of these is a pollination network which shows bees' utilization of different plant species. And you can see that they overlap considerably with the two diurnal species, which are of the same genus, and which essentially tells us they are utilizing the same kinds of resources as the diurnal bees. And what we really don't know so far is what is their role in terms of pollination of wild plants and crop plants and so on. So I'll summarize again by saying that we should be devoting more effort to the study of bees, tropical bees. There are about 20,000 odd species worldwide, and we have a very rich diversity of bees. Yet we know very little, including social bees, which are very charismatic, and they show all these fascinating behaviors. We see that there are various differences, even with the limited amount of work that we know of through research work. We see that there are basic underlying differences, and so therefore this becomes very interesting to study with respect to the tropical bees. And of course, the two temperate bees are not exactly representing the diversity of tropical bees. So with that, I'd like to thank you for this, and also please do catch my two students are here, And one of them is talking about the population genetics of honeybees, and the other is talking about traffic management at hive entrances, how they avoid collisions and so on. So please do catch their talk. Thank you. Yeah. There's no time for questions.
Very good morning, everyone. That was a wonderful start to Econ 2025 by this enlightening talk by Dr. Hema Somanathan. We really thank her for helping all of us here to learn so much about the wonderful life of bees. And now we shall proceed into the very first segment of Econ 2025's technical section. My name is Anuranjan Roy. I will be the host over the next 3 days for 3 sessions of speed talks. We have been dividing the talks into 7 talks per day, so that every day in the morning, all of you get a flavor of brief looks into extensive research done by a variety of very interesting people. And it's my honor to be the host of this particular program. And I would like to begin with the introduction of the first speaker today. The first speaker today is Mr. Uddalok Tathagata. He is a PhD student of the Wildlife Institute of India. His research focuses on the ecology and movement of rhesus macaques in a human-modified landscape. And he has obtained his bachelor's in zoology from Presidency College in Kolkata, plus a master's in forestry. And right now he holds a special interest in urban ecology. And his talk is titled Of Macaques and Men. I would request Uddalok to please come on stage and go through his talk.
Yeah, thank you so much for the introduction and good morning, everyone. On that note from PB Shelley, like arising and unbuilding it again, I would like to introduce you to a species in the very backward of this particular campus, and I'd like to talk about its population density and management scenarios that we have looked into. So going ahead, the amount of agonistic interactions that has taken place in this country with respect to humans and this particular species has always been looked upon from the point of the population rise that has taken place. And some very important studies over a long period of time have suggested some magnanimous rises in particular areas of northern India, which coincides with also a lot of damages due to these particular agonistic interactions that do take place. And this has been a staple of culling, sterilization, translocation of the species. And there have been other methods as well, whether to declare the species as a vermin to finally removing it completely from the Wildlife Protection Act of 1972. So the question arises, if we actually look into it as to how the population density of the species goes through and what are the probable management scenarios that we can look into. So for this particular study, what we did was we looked at anthropogenic mosaic landscape in northern India, and where the macaques are actually synanthropes, where they benefit from their association with humans with close association and also easy access to anthropogenic food. So we divided the 16 square kilometer study area into grids of 1 square kilometer, and we laid out 8 transects, which were sampled each year across 3 seasons, and for every exercise, each transect was sampled temporarily 3 times a day. So overall, across a 3-year period from 2020 to 2023, we had an effort of around 604.5 kilometers in this particular area, where we came across a mean macaque density of around 143 macaques per square kilometer. In comparison to the human population density in this particular area as per the last census, which is somewhere around 2100 people per square kilometer. So an interesting thing to look into it is that because the species is so much under focus with respect to the agonistic interactions that do take place, so a lot of management efforts have already taken place in states like Himachal Pradesh and also Uttarakhand, and with a significant financial investment going into it. So we tried to look into it as to if management interventions do take place, how we look at our study area if this is to go through. So what we did was, based on the density that we arrived from our transect data, so we use that the abundance with respect to the carrying capacity at double that value. And also in this particular region, we have been studying 4 groups which have been habituated since 2018, and we arrived at a mean group size for this. So we use that as well to simulate scenarios if there are interventions being made. So what we came across was quite interesting. So if 80% of intervention is actually made, whereby 70% of the females are actually breeding in the natural condition, within a span of 11 years, the population halves and almost an extinction rate of 50% is achieved. Similarly, if we look at the group scenarios, again, within an 11-year time span, the population halves, but an 100% extinction is achieved. So this was an interesting thing to look at. But when we were actually out there in the field, going by our sightings as to where we were coming across, the number of macaques that we were coming across, so we looked at the cluster hotspots, and this is what emerged. So we found that it was on the edges of particular habitats or the interfaces of particular habitats that the macaques were more prominent, or that is where we had the higher number of sightings or higher number of animals being observed. So this is something to look into, because when we go for a blanket approach in trying to manage a particular species, these are certain intricacies that might be lagging behind. So we kind of propose that whenever a management approach is being taken towards organizing this particular study or area, we need to classify that particular habitat in either rural and urban areas and in forest interface divisions. And followed by that, the approach needs to be either surgical in both males and females in urban and rural areas, but when it comes to a forest interface, an interaction with or an amalgamation of immunocontraceptives could also be utilized. And again, going ahead, when we move ahead into dealing with that particular species, an important thing that can be done is to utilize citizen science programs where photographs of these particular macaques can be used, and we can go ahead and utilize that along with social dynamics and movement data to organize the species. And finally, a word of caution, this particular species was at once abundant in the Southeast Asian countries, but today is an endangered species. So going ahead, we need to be extremely careful about that. And on that note, thank you so much, and thank you for the opportunity.
Thank you, Udalok, for that wonderful study of the animal we are most familiar of at seeing on campus, but rarely know so much about. And thank you for that insight into their behavior and your efforts into that. Next, it is my honor to invite Mr. Gaurav Singh. Gaurav Singh is a PhD student working at the Behavioral Ecology Lab at IISER Mohali. His work involves ecology, bioacoustics, and machine learning. His talk is titled very interestingly, Vocal Fingerprints in a Social Bird. Gaurav Singh.
Yeah, so hello everyone. I'm Gaurav, and I work with Dr. Manjri Jain at IISER Mohali, and today I'm going to talk about my work on vocal individuality in jungle babblers, cooperatively breeding social passerines. So classifying others as mates, kins, or offspring can facilitate cooperation. It can prevent unnecessary fights over territory, ranks, and may also reduce the cost of inbreeding. So such signatures of identity can be actually found across all the animal kingdom, across many acoustically communicating species, but especially these can be utilized when other sense of modalities tend to fail. So motivated by this, we actually ask if there is an individual signature present in the contact call of jungle babbler. These calls are long distance calls and are produced during separation events, so those are likely to encode individual identity. So we actually looked at about 1,000 calls from 41 banded individuals. These calls were taken using handheld recorders from the field, and these were free-ranging birds, so it was quite difficult to get these recordings, especially like from individually banded animals. And still we tried to incorporate as many true replicates as we could to capture more variation. Then each of these calls were annotated, and we extracted different feature sets like MFCCs and bioacoustic features. These were cepstral and spectrotemporal features, and were then further combined to actually classify them using both the raw features and the transformed features. These feature sets were actually put into different sort of classifiers, such as regularized discriminant analysis, support vector machines, and artificial neural networks to cover both linear and nonlinear approaches. Coming to results, when visualizing this this feature space, we saw that over this linear space, there is some degree of separation. However, you can see lots of overlaps over there as well. As compared, when we look at UMAP, which is a nonlinear technique, there we can see more tightly clustered and clearly separated clusters. So this even you can, if you want to focus at one of the individuals, say BOY, which is at the top in the RDA, you can see these data points are kind of dispersed, but here all of these are kind of overlying on top of each other. So this suggests that maybe signatures of individuality can be visualized more in the nonlinear space, and this supports our use of nonlinear classifiers. So we try to evaluate the performances of all the models, one over raw features as well as transformed features, using different metrics. Here we present just only F1 score and ROC values. One can see that at the top, the support vector machine trained on raw features has the best classification performance. But other models such as ANN and RDA weren't lagging behind either. This suggests that the maybe individuality in the calls is more reliable and robust across models. One can also notice that the bottom three, which are actually the transformed features, so models were trained on these features and their classification tends to reduce as compared to the raw features. This basically suggests that some information is getting lost when features are getting transformed or removed. So next we ask if there are certain features which encode individuality the best, and we did permutation importance analysis. We found that other frequency 5%, which contributed most to the individual identity. However, if you look at amplitude-based features, they were like very less informative. So this also suggests that our findings, our classification results are not just an artifact of difference in the recording sessions or background noise. So to conclude, we found that yes, jungle babbler calls contain reliable individual identity, and this can have major implications. Vocal individuality may support group cohesion or social tracking, but behavioral studies needs to be carried out to see if receivers are actually perceiving this information and not. Secondly, combining machine learning models with passive acoustic monitoring can enhance or enable more scalable and non-invasive tracking of individuals, especially in endangered species which requires conservation. On that note, I would welcome collaborations, and if anyone has similar data sets, we can work together. So yeah, I would like to thank ICON for their support and Government of Punjab and my Bell family. To show my gratitude, I will just end this talk with this. Okay, this does not end it. So there was a call which I was supposed to play. This call basically is a song by Lata Mangeshkar which says ki meri awaaz hi pehchan hai, and that is what I was trying to say, that my voice is my identity. Thank you.
Thank you, Gaurav, for that another wonderful talk on jungle babblers. As all of us who have lived in joint families know, there's a lot of personality, and thank you for proving that via science as well. So we look forward to learning more about your efforts and wish you all the best. Next, it is my honor to invite K.P. Akilan. He's a master's student at WII, and he is interested in community ecology and invasive species. The work he'll be presenting was done at Azim Premji University prior to joining his master's, and his work is titled Global Economic Impacts of Invasive Termites. Over to you, Akilan.
Thank you for the introduction. Good morning, everyone. So I'll be talking about the global economic impacts of invasive termites. So just to give a brief introduction, invasive alien species are one of the top 5 drivers of biodiversity loss globally. And often the ecological importance are well studied in comparison to the economic impacts of these species. And economic costs help stakeholders and the common person relate to these costs and also to understand the magnitude of the impacts better. Now, social insects like termites and ants are considered one of the most successful invaders owing to their ability to adapt to a range of environments, their colonial structure, and also their high reproducibility. So having said this, our objective is to calculate the economic impacts of invasive termites and to investigate the distribution of costs in terms of geography, time, taxonomy, and who bears the cost and what type of cost it is. And then finally, to identify the knowledge gaps present in all these areas. So to do this, we perform searches on Google and Google Scholar for all published sources of literature on economic costs of invasive termites. We also contacted researchers working on invasive termites to acquire details about any unpublished details or their funding details. We worked with the InvaCost database, which is a global database collating economic costs of all invasive species. All costs were standardized from multiple different currencies to the US dollars 2017 exchange rates. This figure here shows the pipeline through which our data went through. From the entire InvaCost database, we filtered out the termite data, from which only the observed and high reliable costs were selected, which amounts to about 335 billion US dollars. And of these, we further verified these costs to make sure that they are they are sourceable or their method of estimation is accurate. Now, further, this resulted in a cost of about 627 million US dollars. Now coming to the distribution of costs, this is a Sankey diagram that shows the flow of costs from species, whether it's a potential or observed cost, to the countries incurring the costs and the sectors. So public and social welfare, which relates to human infrastructure, and authorities and stakeholders, which is any government or organizational spending on managing the species. And finally, the cost types, whether it's damage, management, or mixed, which include both damage and management. Now, I would like to point out that you have 6 species represented and 7 countries represented here. Invasive species, there are 28 invasive species globally, of which 42 countries they invade. And you have only this much representation here. And finally, even sectors like agriculture and forestry, which could have potential impacts, are not represented here. Having said this, 335 billion US dollars is the economic burden we are faced with in terms of invasive termites. And this is about 6 billion US dollars per year over the past 57 years. Now, $627 million is the verified cost. This is just to be rigorous in our claims about the cost. Now, if we actually consider potential costs and the low reliable costs, they indicate how high the cost could actually go up to. And I showed that there are many knowledge gaps to be filled in terms of species, geography, and sectors represented. And finally, to conclude, nevertheless, all this is a huge economic burden, yet it is an underestimation of the actual impacts of invasive termites. Thank you.
Thank you, Abgilan, for highlighting that extraordinary cost which has been imposed over so many years, and your study showcasing the impact of invasive termites. Next, I would like to invite on stage Ms. Tamanna Ayappa. She has just graduated with a Bachelor in Biology from Azim Premji University and is looking to pursue her master's. Her talk is titled Tracking the Kaveri's Mahseer.
Should I start?
So good morning. Hi, everyone. My name is Tamanna, and today I'll be presenting a project on a geospatial study where we track the mahseer in the river Kaveri, which is an initiative of the Wildlife Association of South India. So about 1% of the Earth's surface is covered in fresh water, which houses about 50% of all known fish species. And the freshwater ecosystems are among the least studied. And our area of interest, which is the Western Ghats, has an endemism of about 50 to 60%. And each species tends to face different issues for survival, making conservation strategies very complex to address. In this sense, we look to the humpback mahseer predominantly, where there are 16 species of mahseer found globally, 5 of which are endemic to India. And through this study, we tracked the fish in their summer home range in the Kaveri Wildlife Sanctuary for about 6 months last year. And there are multiple characteristics for which we consider this fish. It's a mixed feeder, it's migratory, using multiple habitats throughout its life cycle, and it also is linked to man through angling. So we can compare it to the tiger as a flagship species in the hope that its various needs would cater to the 195 odd species that are part of the Kaveri ecosystem. So in addressing this species extinction threat, we look at two things. One is tracking the movement of the fish through radio telemetry, and then generating habitat maps. The data that we've collected is from last year, between January and June, and this is a visual representation of the sanctuary. So the radio telemetry, we track the fish, about 13 fish, for every hour from 6 a.m. to 5 p.m. every day, and we also log the water quality in the areas where we found them. And we checked the logs, and all the fish that we tracked showed varied habitat dependency, and water levels were the limiting factor of their behavior. So we identified a stretch of river about 3 kilometers called Galibore, which they used as their home range, and we used a drone to capture a map of the physical features of the river, which is the first, which we then used to run a supervised classification, which is the smallest map. And we used the model to then generate a habitat map, which is the last that we see here. So then, to check its accuracy, we ground truthed it, found a 90 to 95% accuracy. And then to check its scalability, we ran the model in 2 other stretches, Bhimeshwari and Mekedatu. This is the preliminary movement data. Essentially, the fish, when the water levels are higher, they're found in the red zones. And when the water levels go down, their mobility is limited, and so they move east towards the green or the yellow zones, where they are able to access lesser habitats in the river. So we are hoping that this data can now be applicable to sites beyond protected areas in river stretches where there is species extinction, and we can start rehabilitation of degraded areas. And WASI has already implemented this data and designated protected stretches of the river in the sanctuary. And this visualization through the habitat maps can help justify our cause to government agencies and inform them on minimum e-flows. So going forward, we are looking to link the habitat maps data as well as the movement data so that we can extract more trends of mahseer behavior. And of course, we have to address the scalability, look at larger areas to consider, as well as tracking more fish. Yeah, so these are some of WASI's collaborators. Thank you. If anyone is interested, feel free to reach out or contact us.
Thank you, Tamanna, for that fascinating talk on the mahseer. We up here in Dehradun have read and frequently see in the Corbett landscape fish like the golden mahseer. It was interesting to hear about the Kaveri mahseer. Next, it's my pleasure to invite Chinmay Sawant on the stage. Chinmay has a keen interest in habitat ecology with a particular focus on marine ecosystems, understanding the ecology of elasmobranch branches and cetaceans. He's particularly passionate about community-based conservation initiatives and is also an awardee of the CCP WWF India government grant.
Thank you, sir. So good morning, all. So fishes we have seen like classified into bony fishes and cartilaginous fishes. So with ever two, these elasmobranch sharks, rays, and skates exhibit a very unique life history traits that makes them very vulnerable to the overfishing and habitat degradation. So globally, 1,199 elasmobranch species has been reported, and in India host around 160 species. So mostly the research, we have able to see that research mostly focused on bycatch and policy data and limited local ecological knowledge based there. So my study, this starts that there is a critical lack of ecological knowledge and about the shark diversity and distribution in India. So with regards to this elasmobranch aggregation, and aggregation is like multiple individuals coming into the one area due to common diver. It's the presence of prey for the feeding areas or and presence of social grouping for the nursery areas or mating systems. So this is a non-social aggregation of grey reef shark in Cagos archipelago, and this social grouping of leopard shark with pups in the southern California. They are coming for the mating purpose. So with regards to this, my study area sites in the Lakshadweep, it's India's smallest island. It's a group of Amandivi, Lakidius and Minicoy, and it's a form of atolls, is a ring-shaped island including a coral and circles with the lagoon. So with this, my objective to understand the species composition of elasmobranch in these selected islands and what's the aggregation sites in this Lakshadweep area. So with regards to this, my methods conclude semi-structured interviews with conducted using convenience sampling. So photographic field guides to identify this elasmobranch by local names and location pinpoint on the map by the fishermen and scuba divers. So data is collected on key aggregation sites, aggregating which species is there and season. So 120 respondents were concluded from the study area from Agatti, Kavarati, and Kadmat Island. So with regards to this, my results, around 24 species of shark is reported, and from that 14 species, these 14 species seen aggregation by the fisher folks and scuba divers. So with regards to shark aggregation, mostly white tip reef shark and black tip shark is shown the maximum aggregation size, and mostly this milk shark and hook-tooth shark, these are like open pelagic coral reef species show very less aggregation seen. So with regards to this, about ray sightings also, around 13 species of rays has been reported, and from that 10 species shows the aggregation. In that we can see white spotted wedgefish and bowmouth guitarfish. These are critically endangered species which not seen by the aggregation, and this ray aggregation mostly dominant by 75 responded by porcupine ray and spotted eagle ray compared to the cowtail stingray. So where are these key aggregation sites of elasmobranch is located? So mostly the 16 sites were identified and in the 4 islands, and mostly it's 8 sites were identified as shark aggregation sites. In this mostly Cheriapani, Valiapani is the uninhabited islands dominant most, and 9 aggregation sites were seen by the ray aggregation sites. So mostly season we have seen its aggregation season is in the post-monsoon area, and pre-monsoon it's very less as compared to that. So with regards to this, my conclusion is to that local ecological, it's just a preliminary data it's like local knowledge of that where these aggregation sites are located and why and how these sharks and rays are coming in that area. So it's like first study to document these aggregation sites in this region, and uninhabited reefs like I have said, this Cheriapani reef, Valiapani and Suheli area lagoon it's seen in the map. It's showing the higher aggregations as compared to the inhabited islands, that highlighting the conservation importance of these species. So way forward, the future surveys will focus on seasonal drone surveys that could be to map the aggregation sites where these areas are there, aggregation sites are there, and long-term study is to be required to gain more precise ecological information of the species where this species has been found. So with regards to acknowledge these people, references, thank you.
Thank you, Chinmay, for a brief look into the pioneering research you are doing on the families of sharks and rays in Lakshadweep. Next, it's my pleasure to invite Mr. Suraj Kumar Das. He has done his master's in wildlife science from North Orissa University, and then he has joined WII in the NMC project National Mission on Himalayan Studies project in the Human-Wildlife Conflict project. Currently, he's working on elephant ecology and human-elephant conflict in Assam. His talk is titled Securing the Corridor of Deosri.
Good morning to all. Thank you for the introduction. My talk will be on a important elephant connectivity zone that is located in the Assam state. Okay. So as we all know, when endangered species have been impacted by habitat loss and fragmentation, then we can go for maintaining ecological corridor and to secure their long-term conservation. So, the Deosri connectivity zone that is located in the Assam state in Bodoland Territory Council area in eastern Himalayan foothill in the, near to the Indo-Bhutan border. So it is a important area for elephant movement as it connects the northeast elephant population to the rest of the India through this area. So as you see, once used to be a contiguous forested habitat, this area has now sink to some riverine patches and some forest patches where elephant used to move. So on a brief overview, the total area, you can see the red box. The total area is approximately 600 square kilometer, located in the Kokrajhar and Chirang district. The major land use type is forest cover, grassland, then agricultural land and riverine habitats. So forest types are mainly moist deciduous forest and alluvial grasslands. And the major community live there is the Bodo tribe and Nepali people. So this is the area we made a brief assessment of the present status of elephant movement in this area through questionnaire survey and remote sensing data analysis. Analyzing the land use land cover map, we can see in between 2017 and 2023, the area has experienced 22% reduction in forest cover, 35% reduction in dried river beds, and 87% increase in human settlements. And the human settlement increases mainly due to the encroachment. So we established elephant movement in this area. You can see the dark green boxes. So these area experience frequent elephant movement, whereas the other area, the red are the not reported area, and the light green and purple color where occasional movements are there. So this is also a transboundary elephant movement zone that elephant move from India to Bhutan through that riverine corridors, riverine habitats. And the I river is the major river here, where elephant prefer to stay inside the riverine grassland and move through the riverine habitats. We also explore which ecological variates through affecting elephant movement, and analyzing the GLM study, we also found that the sand beds are the most favorable ecological variates affecting elephant movement in this area. And coming to the human-elephant conflict scenario in between 2018 and 2023, there were we observed 336 number of crop damage cases and 42 number of house damage cases. And these locations are also overlapped with the elephant movement we have established earlier. Analyzing the conflict patterns, we can see the conflicts are mostly occurring in between the July to December, that is overlap with the paddy season in this landscape. And coming to the elephant involved boat, you can see the graph on the top right corner, the hordes are mostly engaged in the crop damage incident, whereas the solitary bulls and small male groups, they are involved mostly in the human house damage cases. And paddy are the most raided crops there, and semi-permanent house made up of bamboos and other temporary that are mostly affected in the house damage incidents. So the major challenge in this area is the upcoming development, and there is a MOU signed in between India and Bhutan to develop. You can see the left corner map, that the road, that the dotted line is the road. That it is currently in the single lane highway, NH 127, and it is planning to be a 6-lane highway in the next 1 or 2 years. And there is also a parallel railway project that is coming, just aligning to that road. And these are the major threat to this elephant connectivity zone. And in conclusion, I would say this is a very important elephant movement area, and this need to be secured for when planning a development project. The appropriate measures should be taken to make this project sustainable. Thank you.
Thank you, Suraj, for that wonderful insight into all the factors which are required to keep large mammal, which requires a large landscape like the elephant, well and good. Before I go to the last speaker for the day, this is a quick reminder to all of the speakers who have spoken before. So Uddalok, Gaurav, Akilan, Tamanna, Chinmay, and Suraj, that this is the last speed talk session today. So after 5 minutes, all of you will be called on stage, and we will open the floor to questions from the audience. So request all of you to be here at the end of the talk, this next talk, which is the last speed talk for today. It is my honor to introduce Mr. Shailendra Singh Deora. He is currently pursuing his master's in wildlife conservation action at BVER in Pune. He recently interned with the Forgotten Cats of Sainthuk project in Shergaon, Arunachal Pradesh. And his talk is titled Pause and Poop. Over to you.
So hello everyone. Thank you so much, sir, for the introduction. So I'll start with my PPT now. So we all know that threats like roadkills, poaching, habitat destruction and deforestation, and also retaliatory killings have significantly contributed to the decline in leopards population in India. So these threats are especially prominent in places where leopards or any wildlife and human beings share spaces. So this concept of sharing spaces took me to the place called Jawai Bandh, that is in Sumerpur district in Rajasthan. It's a unique landscape famous for its unique coexistence between leopards and human beings. So this area is significantly have got the attention of national and international medias, but lacks some scientific backgrounds. So for my study, I have set 2 objectives. The first one is analyze leopard diet in Jawai Conservation Reserve first and second, and nearby areas. And second was to estimate the abundance of potential prey species in the study area. So for this, I have used 2 complementary approaches. First was the prey density analysis, and the second one is scat analysis. For prey density, I used distance software, and then I calculated the prey abundance in the region. And for scat analysis, all the analysis work was done in RStudio, which includes diet composition, GLM modeling for scat presence, and niche breadth analysis. Then we integrated both the approaches and calculated some electivity indices, which shows the preference of the leopard. Then we prepared a conservation plan for the forest department. So for prey abundance analysis, we have divided our study area into 136 grids. So we can clearly see the area is have high densities of human-associated species. This graph is on a log scale, so the lowest value is way lower than the maximum value. For scat analysis, we have done a systematic surveys ensuring the leopard presence also, and we have collected the scat on the basis of physical attributes like smell, odor, and shape. Then we perform diet composition analysis, which includes frequency of occurrence and relative occurrence. So for that, we have examined hair samples from the scat and for identification of the prey using their cuticular characteristics. These are some examples of the species we got. So for the relative abundance, it shows a clear pattern that leopards rely heavily on the domestic prey, especially goats in the human-dominated landscape. Yet they also consistently consume species like mongoose, suggesting that they still seek out wild prey when possible. So to know whether leopards are preying on what is available, we did a Spearman's correlation analysis. This shows that prey availability strongly influences the leopard foraging patterns. Then for the niche breadth analysis, we have used Shannon index to get the dietary diversity of the leopard diet, which comes to be a moderate values. And further to quantify this flexibility of the diet, we have done a Levin's niche breadth index, which shows whether a leopard is behaving like a generalist species or a specialist species. These values, that standardized value shows that if it's 0.5 to 1, it shows that it's on the side of general species. So it's moderately generalist, but still it prefers some species by the preference. So finally, we did a prey preference analysis using Evelev's and Jacob's index. It interestingly shows that leopards are actually preferring wild prey, but they have to turn to domestic prey because of their high availability. So for discussion, I'd like to say that this area have high livestock dependency. Livestock may offer lower hunting risk and energy cost, and also high number of livestock reduces the competition of the predators. Hence, this could be the reason for the high densities of leopard in the Jawaiban region. Yet leopards exhibit a strong preference for wild pig and porcupine-like species, despite their relatively low abundance. These prey species likely provide a higher caloric return, making them energetically profitable targets. Also, leopards actively avoid hunting prey like nilgais and monitor lizards, most probably due to their size, defensive mechanism, and difficulty in subduing. So this all reflects to the concept called optimal foraging theory. So livestock offers an easier and more predictable food source, while wild prey, despite being higher in nutritional value, comes with a greater hunting cost. So for the conservation implication part, so we know that Java is famous for this coexistence, but if this thing keeps going on, the Java is going to lose the tag of this harmony between leopards and human beings and turn into a high conflict zone. Also, this preference of wild prey is going to again create a ripple effect of hunting more on wild prey, then eventually decreasing the wild prey, already dwindling wild prey population, and also then the leopards have to prey on domestic animals more, then it will again increase the human wildlife conflicts in the region. So these are some scenarios after. So this news was from 21st March 2025. This was the first human fatality in the region. This area is among those areas which have no conflict from the leopards in the world. Actually, 1 or 2 places are like this is there. So thank you so much.
Thank you, Shailendra, for that very informative talk on the Jawai landscape. We've seen some amazing wildlife photographers post from that particular place. And as someone interested in heritage, I find that the balance between nature and culture is quite important and fascinating to study. So now we have, in the true nature of Speed Talks, we have speeded through 7 talks. And now I'd like to invite all those presenters of today. So Uddalok, Gaurav, Akilan, Tamanna, Chinmay, and Suraj back on stage. I would request you to line up here, and for the audience, we are almost on schedule. Maybe we have till about 20 odd minutes for questions. So if anyone would like to ask a question, I will request you to just raise a hand first, and then we can direct it to the, you guys can come in the center if you want. So whoever gets a question can come to the mic and speak. Right, so amongst the audience, do we have a few questions for any of the presenters? Please, whenever you are speaking, introduce and try to mention the topic of the presenter if you've forgotten the name, right? So please, any audience questions? Okay, right. Qureshi sir, maybe we'll begin with, Qureshi sir, we can begin with you. Qureshi sir.
Javai presentation. Sorry, I forgot your name. Shailendra. So Shailendra, just is an interesting area, but just I want to comment on that. That Javai experiment exists because of power dynamics of caste and money. So that experiment is stitched more artificial. What's your opinion on that?
So I feel like the youth in Javai is shifting towards this, we can say, unsustainable views of practices of ecotourism in the sector. So because of these, we had an equitable benefit sharing concept before in the region, in the communities, but now this youth are starting their own businesses in the region, and they are leaving the previous resorts and all these places. So they are like, we'll start our own businesses and all, and offering a cheaper prices, which is like affecting the gypsy, like it's increasing the gypsy population in the region, and there is no way to control this population because this area is fully controlled by the communities. So I think targeting that part of the population of that region would make some effect in this region. Thank you, sir.
Thank you, Kamal sir. Thank you, Shailendra. Do we have questions? For those of you in the mic, you can switch your mic on and speak. For those of you behind, we will try to get a mic for you, but as of now, if you can just raise your hand and maybe come forward to one of the mics and ask a question. Yeah, please, for those who are seated behind, can you just come up to one of the mics and ask a question? Or can we have someone pass a mic around at the in the rear rows? Because there's a question there. Okay, there. Right mein. On the far right, if I may say so.
In the meantime, can I quickly ask something?
Sure, ma'am, go ahead. Oh,
again,
sorry, ma'am,
Shailendra,
yes.
You said the wild prey are the more nutritional. How do you know? Has it been tested?
Ma'am, I have mentioned some papers which shows that there are higher caloric returns for the wild pigs and this wild species like nilgais and all. I'll mention those papers. I can forward you the.
So in that case, one of the major predictions of the optimal foraging theory is that the choice of prey will depend on the availability of the more profitable prey, as opposed to one which is easy. But if it is available, why not just eat? That's also has been addressed in the optimal foraging theory, right? So if there are so many livestock available anyway, and if it encounters it first, it will anyway consume it, right? Unless the handling time is much more. So then...
So they are actually relying on domestic animal more, and also wild prey species way less than the domestic species. And so if this wild prey species goes down, then they will turn to domestic prey more and more, and it will create a kind of ripple effect. So then the conflict will increase. So this tolerance will erode by time. So nobody will take a loss of 1, 2, or even more the livestock of their own.
What I meant is it's unsurprising. You encounter it first, you will consume it unless the handling time is greater, no?
But as we can see, the leopards are preferring the wild prey. So maybe it can be because of the this higher caloric returns. I'm not sure. I've not done research on that part, so thank you.
Yeah, thank you, Shailendra, once more, and thank you, ma'am, for the question. Can I request the concerned person to maybe come up front? Sorry about that. The mic seems to be a little malfunctioning today.
Hi, my question is for Gaurav. Yeah. So for testing individuality in calls, do you think you also need to do some experiments on the same individuals again, meaning like repeatability? Because it could just be like you're looking for a signal in the individual call, but you also need to say that it's, you know, consistent in the same individual, right?
So actually we did take true replicates. Like these calls have been taken across 4 years. So we have we don't have that many calls, but still we have taken many calls from say for an individual, at least 3 calls are there which are across from different years. So still we have seen them being clustered together. So this infers that there may be still it's quite possible that identity is not consistent across years, but still there may be some features which are staying consistent. That's why we are still seeing them together in the clusters.
Thanks. Can I ask one more question? Yeah. This is for Shailendra. So when you say that the wild prey populations can be increased, right? So wouldn't that also lead to increase in the predator populations eventually, and that might in turn lead to increased conflict?
So that is what my thesis is about. So actually wild prey population is decreasing by time because they are preferring the wild prey. So my discussion was that if they keep relying on this easy prey, and also the population in Javai region of leopards is actually very high. So it's around 40 to 50 individuals in a 350 square kilometer area. So it's already high and it's easy for them to prey on these domestic animals. So that could be a reason that this population of leopards is already high. And the thing with big cats is if they have a standard range. So if one even goes out, the another will fill the space automatically. We don't have to do anything in that scenario.
No, but as an intervention, if you increase wild prey populations, then that will also lead to an increase in the predator populations, the leopard populations, right?
So interpredatory conflicts you are mentioning. So
no, I'm referring to a numerical response. So leopard numbers will also go up, right? So this is what I'm trying to say is that it's a tricky problem to solve.
So actually we need a wholesome approach. So it's not that we are significantly increasing the wild prey population. We are just increasing up to the point that they don't have to rely on domestic prey as much as. So earlier also they were staying in this place only, in that region only. But that time there is a possibility that there was a balance between wild prey and domestic prey. So now that the wild prey is decreasing and domestic animals are increasing in that region, and also this human pressure is also more, then they are choosing to rely on domestic prey more.
Thanks.
Thank you. Can I ask a question to Chinmay?
Yes.
Chinmay, you said your analysis was based on local ecological knowledge, LEK, and your interview numbers were 120. Some of them were tour operators and others were traditional fishermen. Don't you think your sample size and your work will be biased because the fishing in Lakshadweep, mostly pole fishing for tuna, and also the tour operators operate in very definite areas.
Sorry, it's scuba divers, not
yeah, scuba divers. Yeah, they operate in extremely. Scuba divers only operate in extremely small areas. So I think the analysis, you have 24 different species recorded based on your 120 interviews. Do you have long-term plans of your survey to be extended into wider areas, or you are restricting yourself to only LEK? That's my question.
Yes, sir. Thank you, sir, for the question. So, maximum, sir, 2 months I got to surveyed in Lakshadweep from February to April maximum. So in that March month is not. So it's like, yes, sir, you told like it's very less sample. So just 100 fishermen has taken and 20 scuba divers. So yes, for this is like just preliminary to know which is the sites areas aggregation, and after that drone surveys we can use, and weighted remote underwater cameras also we can use for long term to see exactly like which are the places. And also it's restricted to just 4 islands like Kavaratti, Kadmat, Bitra and Agatti. So it's having 10 islands more. So I think we have to cover like.
Do you have plans to also go into uninhabited islands? Because
yes, sir.
Okay.
Yes, sir.
Yeah.
In uninhabited islands also I went like Cheryapani and Valyapani Reef. That is like just one survey only I did, because it's very logistically difficult to go that area. So like it's need like long term project to get more intention to like where which are the places here.
Thank you, Chaudhary sir, for the question, and thank you, Chinmay, for the answer. Do we have any other questions in the audience? Okay, I can see Sutirtha sir. Sutirtha sir.
Hi, there is a question for Udalak. Udalak, the demographic models that you've used to test the effectiveness of planned interventions, do they also incorporate the density dependence in reproductive rates? That means if 80% of the females are sterilized, whether the remaining 20% will increase their reproductive rate?
Yeah, we did consider that. So an interesting fact here in this particular landscape that we have observed, if we go by the previous published literature on long-term studies, so there the females take about a 2 years gap to reproduce, for the next offspring that comes in. But in our case, because we have identified female individuals and stuff, so we came across multiple females who used to give birth every year. So even with the gestation period of only about 5.5 months, they were able to bridge that gap in trying to reproduce because of the availability of resources and the lack of predators in this particular area. Thank you.
I have a question for Kaveri Mahseer. Just for information, what are the major threats for mahseer in that area? A and B, though you've not done, you told about daytime. Do you think that there will be a nighttime differences just for your experience working in that area? In habitat use.
Yeah, so firstly, you want to know about the threats, right? So one of them, the major threat is that the KRS dam, right? There are 2 dams, KRS and Kabini primarily. And the water levels keep, I can give you a story actually. So one of the females that we were observing, Mara, we lost her for about a month while we were doing the logging of our data. And this is because there was a drought period last year and they opened the KRS. So water levels went up and we assumed that she went upstream. And they went down in about 6 hours after like 48 hours of leaving the dams open. And she died. She got trapped in a side channel and she died. So that's one. The other is there are about 4 breeding populations in the stretch that we are looking at. And since they are constrained in like certain areas of the river, if due to logging, if there's a landslide that happens, then the entire population will be wiped out. So that's 2 threats, logging and the dams. And secondly, sorry, what was the second question?
We've not done study, but in nighttime habitat use, you'll see any difference, whatever data you have.
Yeah, so like I said, we stopped the observations at around 5:30 p.m. every day because we found that the signal that we're using has a range of about 500 meters. And we found that the fish would no longer be trackable, so we think that they go into the deeper pools or under rocks in the river, so we can't track them. Yeah. Yeah. Okay, thank you. Yeah. So for Vasi's been working in the sanctuary for about 50 years now, and there have been major angling camps which they were banned, and because the mahseer is on the red list now, so it's endangered. So yeah, we also have, there was massive poaching that was happening because of its size. Yeah. Okay, thank you.
Thank you, Tamanna. Thank you, sirs, who had asked the question. Do we have any other questions right now? I think we are done for questions. Thank you so much for to all of you who sat through. A big hand for the people who spoke. Before wrapping up, I'd just like to thank the rapporteurs Shivam and Radhika, who have been working a lot in the background to make this event happen. And I don't know if Shivam, if you want to make a quick comment. You good? Okay, okay. So Shivam is good for today. He will maybe wrap up on the third day. So tomorrow again we have speed talks. The time would be from 9:45 onwards. So those of you who spoke today would request you to be here tomorrow to see your competition and also support them. So we'd like to have you over here and look forward to more questions tomorrow. So thank you, and let's break for tea.
check, hello, 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 check, hello, 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 check, hello, hello, check, hello. Hello, check, hello. Hello, check, hello, hello. Hello, check, hello. Hello, check, hello. Hello, check, hello. Hello, hello, check, hello. Hello, check, hello, hello. Hello. Hello, check. Hello, hello. Hello, hello. Hello, hello, check, hello, 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 hello. hello, hello, hello.